6 kilometers down in total darkness, there's a squid with eyes roughly the size of a soccer ball. Nearby, crabs stretch nearly 4 meters from claw tip to claw tip. Somewhere in that same crushing blackness, a jellyfish trails ribbon-like arms as long as a city bus. None of this is exaggeration, and none of it is especially rare once you know where to look. The deep ocean, the coldest, darkest, most food-starved environment on the planet, has spent millions of years quietly building some of the largest animals alive today. That's a genuine paradox, and it's one that puzzled biologists for more than a century. Less than 1% of the organic material produced near the ocean's sunlit surface ever survives the long fall to the seafloor. Resources down there should be scarce enough to punish anything large, favoring small, efficient scavengers instead, and yet the opposite keeps happening. In 2025 alone, 3 separate breakthroughs, a live sighting, a genome sequencing effort, and a biomechanical study finally gave scientists a coherent explanation for why the deep sea keeps producing giants instead of dwarfs. Here's what they found. The starting point for understanding deep sea gigantism isn't biology at all. It's basic geometry, and it dates back to a German biologist named Bergmann, who noticed something in 1847 that still holds up today. Animals living in colder climates tend to be larger than closely related animals living in warmer ones. The explanation comes down to the relationship between surface area and volume. Take a cube, 1 centimeter on each side, and it has 6 square centimeters of surface area holding in 1 cubic centimeter of volume. Double the length of each side to 2 centimeters, and the volume increases 8-fold, while the surface area only increases 4-fold. The bigger an object gets, the smaller its surface area becomes relative to everything packed inside it. For a living animal, that ratio directly determines how quickly body heat escapes into a cold environment. A larger body has proportionally less exposed surface through which to lose heat, and a slower rate of heat loss means a slower, more economical metabolism that can coast for longer between meals. This single principle, now known as Bergmann's rule, turns out to explain an enormous amount of what happens in the deep ocean, where water temperatures below roughly 1,000 meters typically hover just a couple of degrees above freezing year-round. Cold-blooded animals down there still generate some metabolic heat as a byproduct of ordinary cellular activity, and a larger body simply holds onto that heat more efficiently, letting its internal engine idle at a fraction of the rate a smaller relatives would need to survive. Down here, growing large is an excess. It's an energy-saving strategy, arguably the single most important one available. The giant isopod is one of the clearest living demonstrations of that principle in action. Related to the small woodlice you might find under a rock in your backyard, some giant isopods reach over 30 centimeters in length and close to a kilogram in weight, roughly 50 times the mass of their shallow water cousins. They're built like armored tanks, with 14 legs, 2 compound eyes made of thousands of individual lenses, and a mouth of jagged plates designed to shred whatever falls into reach. What they're famous for, though, is how little they need to eat. When a dead whale, tuna, or other large carcass drifts down from above, giant isopods will gorge themselves until they're physically too swollen to move, a state researchers have somewhat dryly termed post-feeding immobility. One individual kept in a Japanese aquarium famously went more than 5 years without eating a single meal, apparently by choice rather than illness, simply drawing down a metabolism that barely needed replenishing. That's Bergmann's rule playing out almost perfectly. A body large enough to store substantial fat reserves, combined with a surface area to volume ratio, efficient enough to make those reserves last for years rather than weeks. Bergmann's rule explains why bigger is metabolically advantageous in cold water, but it doesn't fully explain how an animal actually achieves that size at the genetic level, and for one of the deep sea's true giants, the answer turned out to be written directly into its DNA. Alicella gigantea is the world's largest amphipod, a shrimp-like crustacean that, in shallow coastal waters, typically grows no larger than a grain of rice. In the deep trenches of the Pacific, some 7 kilometers down, Alicella instead reaches over 30 centimeters in length, roughly 50 times the size of its shallow water relatives. When researchers sequenced its genome in 2019, they found something that startled even the scientists running the analysis. At roughly 34 gigabases, Alicella's genome dwarfed every other deep-sea amphipod studied by a factor of nearly 9, making it one of the largest crustacean genomes ever recorded. Crucially, across the animals studied, genome size tracked directly with body size. Larger genetic blueprints correlated with larger bodies, as those size were quite literally written into the instructions before the animal ever began developing. A follow-up study in 2021 dug further into what that oversized genome was actually doing. Researchers compared Alicella's active gene expression against a much smaller relative and found clear evidence of positive evolutionary selection acting on genes that regulate cell division and mass accumulation, genes that were being actively pushed generation after generation toward greater and greater growth. In most animals, The biological signal to grow eventually switches off once adult size is reached. In Alicella, that switch appears to simply never fully engage, a condition biologists call indeterminate growth, meaning the animal keeps slowly adding size for as long as it survives, with no hard-coded ceiling built into its biology. At the ocean's surface, carrying a genome that large would be a serious liability. Bigger genomes take longer and cost more energy to copy every time a cell divides, a real disadvantage in a fast, competitive shallow water ecosystem. But 7 kilometers down, where competition is sparse and metabolic speed barely matters, that same expensive genome becomes an asset rather than a burden. One of the clearest examples yet of a genetic trait that only makes evolutionary sense in the specific context of the deep sea. A different, equally strange growth mechanism shows up in one of the ocean's oddest animals, the Antarctic sun starfish, which can carry as many as 50 arms and reach 60 centimeters across, living in water that sits barely above freezing all year long. In 2023, researchers mapping the genes that instruct developing cells which body part to become made a striking discovery about starfish anatomy in general. Essentially, the entire animal, arms and all, corresponds developmentally to what would be the head region in most other animal body plans. It's an animal that is, in a very real sense, mostly head, one that evolved the ability to crawl around and feed by ejecting its own stomach out through its mouth to digest food externally before drawing everything back inside. The size question, though, comes down to what happens inside individual cells, rather than how many cells there are. There are two fundamentally different ways to build a larger animal. Pack in more cells, which is roughly what elephants do relative to mice, since an elephant's cells are similar in size to a mouse's, just vastly more numerous, or make each individual cell bigger. Inside every dividing cell, two processes run in a kind of race. The cell steadily builds up mass by producing proteins, while a separate signal periodically triggers division, splitting the cell in two and resetting its size back down. In warm water, those two processes stay roughly in step, keeping cell size fairly constant across generations. In water, hovering near freezing, though, the chemistry shifts. Protein synthesis keeps running at close to its usual pace, but the signal that triggers division slows down considerably, meaning cells keep accumulating mass for longer before finally splitting. The result is a starfish built from oversized building blocks, and a wall built from oversized bricks ends up an oversized wall. Move from crustaceans and starfish to arthropods more broadly, and a different physical constraint comes into play: how oxygen actually gets delivered around the body. Every insect, spider, and crab shares the same basic circulatory design, an open system in which blood, more precisely a fluid called hemolymph, sloshes freely through the body cavity rather than traveling through a closed network of veins and arteries. It's a workable design at small scale, but it puts a hard ceiling on how large an animal built this way can grow on land, since diffusion alone becomes too slow to supply oxygen efficiently once a body crosses a certain size. It's part of why the largest living land insect today, New Zealand's giant weta, weighs barely more than a golf ball, even though fossil dragonflies from roughly 300 million years ago, when atmospheric oxygen levels ran nearly double today's concentration, achieved wingspans of 70 centimeters using that exact same open circulatory plan. Cold seawater offers a very different kind of oxygen boost. Water, at around 10 degrees Celsius, holds roughly 40% more dissolved oxygen than warm surface water. And off the coast of Japan, a deep cold current has been sweeping steadily across the same stretch of seafloor for millions of years, long enough for local evolution to fully capitalize on that extra oxygen supply. The result is the Japanese spider crab, known locally as Takaashigani, or tall legs crab, the largest living arthropod on Earth, with a leg span stretching nearly 4 meters tip to tip and a body weighing up to 19 kilograms. The same open circulatory system that caps insect size on land becomes far less limiting here, because cold water simultaneously slows every cell's metabolic demand for oxygen, while increasing how much oxygen is actually available to meet that reduced demand. As a long-lived decapod crustacean, the Japanese spider crab also keeps growing indeterminately for as long as it survives, which, for some individuals, may mean a lifespan stretching past a century, one slow incremental molt at a time. Not every deep-sea giant grew large by accumulating mass efficiently. Some solved the energy problem by nearly eliminating activity altogether, and the goblin shark is probably the clearest example. Living around 1,000 meters down in the twilight zone, the goblin shark carries a soft, weakly calcified skeleton, poor eyesight, and an oddly flabby, unstreamlined body that would make it an easy target on a coral reef. Instead, it's survived essentially unchanged for over 125 million years, a lineage that predates the extinction of the dinosaurs by tens of millions of years. Its strategy is almost the opposite of speed. It hangs motionless in the water, detecting the faint electrical fields. Every living animal generates through muscle activity via hundreds of specialized receptors packed into its snout. When a target comes within range, Two pairs of elastic ligaments release a jaw mechanism that had been held cocked under the skull like a loaded spring, launching the entire jaw forward at over 3 meters per second in less than a quarter of a second, first captured on film in 2016. The logic here is pure energy accounting. At 1,000 meters down, sustained chases burned far more calories than most available prey could ever repay. By waiting motionless and striking only when success is close to guaranteed, the goblin shark spends almost nothing on the vast majority of its existence, tolerating being slow and unremarkable everywhere except in the one split-second action that actually matters. Depth brings a second major physical constraint beyond cold and scarcity. Pressure. At sea level, roughly 1 kilogram bears down on every square centimeter of skin. Add another kilogram for every 10 meters of additional depth, and by 6,000 meters down, that pressure exceeds 600 kilograms concentrated on an area the size of a thumbnail, more than enough to implode rigid engineered structures in a fraction of a second. The giant phantom jelly, Stygiommedusa gigantea, thrives at exactly those depths by having essentially nothing rigid for that pressure to crush. Its bell reaches roughly a meter across, trailing 4 broad ribbon-like arms that can stretch 10 meters or more, structures built almost entirely from water, with no shell, no bone, no gas-filled cavity of any kind for external pressure to collapse. Because its tissue is nearly incompressible water through and through, internal and external pressure equalize instantly, leaving nothing rigid enough to fail. Where an engineered submersible would buckle, this animal simply has nothing to buckle in the first place. A strategy shared broadly across the deep sea's soft-bodied giants. The oarfish takes a related approach to survival, treating most of its own body as replaceable. The longest bony fish alive, oarfish have been reliably measured at up to 8 meters, with credible but unconfirmed reports stretching considerably longer, in part because almost every oarfish ever recovered by humans was already dead, dying, or missing significant portions of its tail by the time it was examined. Roughly 3/4 of an oarfish's body length is tail, while every organ essential to survival, heart, digestive system, reproductive tissue, is packed into the front quarter. Oarfish are known to deliberately shed portions of their own tail throughout their lives, sometimes losing more than half its length. A behavior scientists still don't fully understand, given that, unlike a lizard shedding its tail to escape a predator's grip, the oarfish has no confirmed natural predators, and the lost tissue never regenerates. As bony fish, oarfish share the same indeterminate growth pattern seen throughout much of the deep sea's giant lineup. No hard-coded signal ever tells the body to stop adding length, meaning nobody has ever actually confirmed the species' true maximum size. For a full century after it was first identified from fragments recovered inside a sperm whale's stomach, the colossal squid, the heaviest invertebrate on the planet, had never once been captured alive on camera in its natural habitat. That changed on March 9, 2025, when scientists aboard the Schmidt Ocean Institute's research vessel, Falkor, operating the remotely operated vehicle, Sebastian, near the South Sandwich Islands in the South Atlantic, filmed a juvenile Mesonychoteuthis hamiltoni drifting through the water column roughly 600 meters down. The individual filmed was barely 30 centimeters long, nearly transparent, in a striking contrast to the fully grown adults, which can stretch up to 7 meters and weigh over 500 kilograms, making them, size for size, considerably heavier body than the more commonly documented giant squid. Adult colossal squid carry the largest eyes of any known living animal, up to 27 centimeters across, roughly the size of a soccer ball, an adaptation for detecting the faintest traces of bioluminescent light in permanent darkness. Their blood runs blue rather than red, using a copper-based molecule called hemocyanin instead of iron-based hemoglobin, a chemistry that functions more efficiently in cold, low-oxygen water. Based on growth ring analysis of small internal balance organs called statoliths, which lay down daily growth layers much like tree rings, researchers estimate the species may reach its full adult size in as little as 1 to 2 years, an extraordinarily fast growth rate compared to most of the other long-lived giants found at similar depths, and one that still isn't fully explained at the genetic level, since the colossal squid's own genome has yet to be sequenced. Before any of the last few years' breakthroughs, most of what scientists knew about deep-sea giants came from a distinctly unglamorous source: dead specimens, washed ashore, tangled in fishing nets, or recovered from the stomachs of the whales that hunt them. That's a fundamentally different way of studying an animal than watching it move, feed, and behave in its natural environment, and it left enormous gaps in even basic questions, like exactly how large a species could grow, how long it lived, or how it actually captured prey. The colossal squid is a good illustration of just how thin that evidence base was. From its formal identification in 1925 until the 2025 sighting near the South Sandwich Islands, essentially everything scientists knew about the species came from a small number of adult specimens accidentally caught in commercial fishing operations, plus fragments recovered from sperm whale stomachs, the squid's only significant natural predator. Estimates of its maximum size, its lifespan, even basic questions about how its enormous eyes functioned in near total darkness were built almost entirely from postmortem specimens, rather than any direct behavioral observation. The same pattern holds for the oarfish, where, as previously noted, nearly every specimen ever recovered arrived already dead, dying, or with a significant portion of its tail missing, meaning claims about the species' true maximum length have rested on inference and extrapolation rather than direct measurement of a living, fully intact individual. This mattered for more than scientific completeness. Conservation planning, population estimates, and even basic questions about a species' vulnerability to fishing pressure or deep-sea mining all depend on knowing roughly how many individuals exist, how quickly they reproduce, and how long they live. When the underlying data comes almost entirely from a scattering of dead specimens collected over a century, those estimates carry enormous uncertainty, and in some cases they may simply be wrong in ways nobody can currently detect. The shift away from fragment-based guesswork toward direct observation and genetic sequencing didn't happen gradually. It happened in a tight cluster, largely within the past 5 to 10 years, and the reasons come down to a handful of overlapping technological developments arriving at roughly the same time. Remotely operated vehicles, ROVs, capable of operating routinely at depths of several thousand meters, have become dramatically more capable and more widely available to research institutions that couldn't previously afford dedicated deep-sea exploration programs. The Schmidt Ocean Institute's Sebastian, the ROV responsible for the first colossal squid sighting, is a fairly recent addition to a small but growing fleet of vehicles built specifically for this kind of exploratory science rather than commercial or military use, equipped with camera systems sensitive enough to capture footage in near total darkness without disturbing bioluminescent or light-sensitive animals in the process. Genomic sequencing costs have also fallen dramatically over roughly the same period, to the point where sequencing an entire crustacean genome, the kind of analysis that revealed Alashella gigantea's unusually oversized genetic blueprint, is now realistic for a single research team rather than requiring the resources of a major national genome center. That drop in cost has made comparative genomics across multiple deep-sea species newly practical, allowing researchers to actually test hypotheses about genome size and body size across a range of related animals, rather than examining a single specimen in isolation. And there's a simpler factor, too. Sustained institutional interest and funding specifically directed at deep-sea exploration, exemplified by organizations like the Schmidt Ocean Institute and the broader Ocean Census Initiative, which coordinates expeditions across the world's least explored ocean regions. Sustained, well-funded, purpose-built deep-sea expeditions are still a relatively recent development, and each one adds directly to what remains an extremely small existing catalog of confirmed live sightings for the ocean's largest and strangest animals. It's worth being direct about how much of this picture still rests on inference rather than settled fact, because the honest state of deep-sea biology is one of genuine ongoing uncertainty rather than a fully solved puzzle. The true maximum size of the oarfish remains genuinely unknown, since, as noted, most recovered specimens are missing tail length by the time they're measured, and credible eyewitness reports of considerably longer individuals have never been paired with a verified, intact specimen. Alashella gigantea's link between genome size and body size, while striking, is based on comparing a relatively small number of amphipod species, and researchers are still working out whether that same genome-to-size relationship holds true across other unrelated deep-sea giants, or whether it's a pattern specific to this particular group of crustaceans. The mechanism behind the oarfish's deliberate tail-shedding behavior remains genuinely unexplained, with researchers able to rule out some hypotheses, like predator evasion, given the species' apparent lack of natural predators, without yet having a confirmed alternative explanation to replace it. Even the colossal squid, despite its landmark 2025 sighting, remains almost entirely unstudied at the genetic level. No colossal squid genome has been sequenced, which means researchers still don't know whether its remarkably fast growth rate, reaching full adult size in as little as 1 to 2 years, relies on the same kind of RNA editing machinery documented in its cephalopod relatives, or on some entirely different genetic mechanism unique to the species. That's a significant gap, given how central genetic sequencing has been to explaining size and growth in nearly every other deep-sea giant discussed here. None of this uncertainty undermines what has been established. It simply reflects how early this particular field of research still is, and how much of the deep ocean, by some estimates more than 80% of the seafloor, remains completely unmapped, let alone directly observed, continues to sit entirely outside the reach of any study conducted so far. It's easy to treat deep-sea giants as pure novelty, strange animals living far enough away that they'll never intersect with ordinary human concerns. But several of the biological tricks these animals rely on have already started crossing over into medical and scientific research with direct human relevance. Marine-derived compounds have already produced real pharmaceutical breakthroughs. A bacterium isolated from deep-sea sediment near the Bahamas in 1990 yielded a compound now in late-stage clinical trials for glioblastoma, one of the most difficult cancers to treat, specifically because it's capable of crossing the blood-brain barrier that blocks most existing cancer drugs. More broadly, marine organisms have already contributed a dozen FDA-approved cancer treatments, with several dozen more compounds currently working through clinical trials. The giant isopods' multi-year fasting tolerance has drawn interest from researchers studying metabolic suppression, with potential relevance to organ preservation techniques and even the physiological challenges of extended human spaceflight, where minimizing resource consumption over long durations is a serious engineering constraint. And some cephalopod relatives of the colossal squid display an unusually high rate of RNA editing, correcting or altering their own genetic instructions after transcription far more extensively than nearly any other animal group studied, a mechanism some researchers are now investigating for potential insight into correcting the kind of corrupted cellular instructions that drive uncontrolled growth in human cancers. What ties every one of these animals together isn't a single mechanism. It's a shared set of physical pressures, extreme cold, crushing depth, and near total food scarcity that happen to reward exactly the traits that would be liabilities anywhere else on the planet. Slow metabolism, oversized cells, genomes that never stop signaling growth, bodies built from water instead of bone. None of it works at the surface, and all of it works remarkably well 6 kilometers down. Most of these discoveries, the colossal squid's first video, Alishella's sequenced genome, the biomechanics behind the goblin shark strike, have only come together within roughly the past 5 years, and largely because deep-sea exploration technology, remotely operated vehicles, high-resolution cameras, and genomic sequencing has only recently become capable of reaching and documenting these animals in anything resembling their natural state. Given how much of the deep ocean floor remains entirely unmapped, and how many of these species were only ever known from washed-up carcasses or whale stomach fragments until just the last few years, there's good reason to think the largest and strangest deep-sea giants still haven't been found at all. That last point deserves to sit for a moment rather than function purely as a closing flourish. Every single animal covered here, the isopod, the amphipod, the starfish, the spider crab, the goblin shark, the phantom jelly, the oarfish, the colossal squid, was already known to science well before the most recent wave of research began. What changed wasn't the discovery of new species, so much as a genuinely new ability to observe already known animals doing what they actually do, alive in their own environment, rather than reconstructing their biology secondhand from whatever fragments happen to wash ashore or turn up in a net. That distinction matters, because it suggests the next major shift in understanding deep-sea gigantism probably won't come from finding an entirely new kind of giant. It will come from finally getting a clear, direct look at giants researchers have known about for decades, and discovering that the fragments never told the whole story to begin with.